Today we're getting started with some microscope play. I finally pulled my microscope out of storage. Want to do a couple audio tests first. Seems like it's working well. Hello, Chloe. Yeah, so first thing we're gonna do before we get going is we're just gonna start cleaning the microscope.
What are you doing?
Well, I'm gonna clean the microscope and then I'm gonna look through some of the water. You want to look at some real quick? All right, you know what? We're gonna do a quick version. We're just gonna get started like ASAP. So my microscope, OMAX, 40X to 2500X is pretty sweet, and I have a little digital adapter so I can see it on my computer. I'm gonna start by turning on the light on the bottom.
Can I do it?
Yeah, here you go. Oh, gonna turn the light on.
No, I want to do everything.
I'm gonna do everything. All right. Well, first, one thing at a time. So, what are you doing right now? You wanna tell us? Are you opening the jar of lake water? Yeah, this one's a little hard. It has a safety lock on it, so you actually have to push down before you open. Hey, go ahead and tell mom. Tell mom we're gonna look at the microscope for a minute. We went to the lake today. Oh, we actually went to a park that has a lake, and then we decided to take samples of the lake water alongside some yard water where we added water. Okay, so we're gonna do it now. So this dropper's broke. Do we have any more droppers? Do we have any more water droppers? I can use this one. It's fine, but it's like it's a little broken.
We do have
Right.
more, but that one is kind of broken. When you get stuff, when you put water, when you put water out, it will spit out water from here.
Yeah, exactly. There's a hole in the side. See, if you go too fast, we can only suck up a little bit of water.
And they do.
Yeah, you want to get the slide cover. So we have our slide. So you can't see it, what we're doing. I should put this in front of the camera. But we have a slide, and we applied a drop of water from the lake, and it has just a bunch of soot and dirt. Oh, let's see. We already have one open, Chloe. We should have one. Yeah, that one. I think I have another slide cover somewhere. What's up, D-Money? Yeah, I got some milk tea. Fresh? No, it was fresh like 6 hours ago, and then I I got distracted. Oh, perfect. You don't have to push it down. You just kind of lay it on top. So it looks like we have some, you see that big black chunk in there?
Mm-hmm.
And it's not letting the slide cover go all the way down. Normally I like to get the slide cover to form a slightly better seal than that. And we'll start up at the lowest magnification, 4X. And let's just...
Is this 4X?
Yeah.
Like this.
So this is 40X.
Is there inside?
Yeah, so
And let me
make sure everything looks good. So don't move the microphone. You can come over here in front instead. Here, why don't you sit down? You can be the scientist.
How can I do this then?
So basically, just move these two, and then look at that screen right there.
I forgot about that one, but I don't forget about this one. This one is just for close, right?
Yeah. Let's see.
I can look.
And,
I can look.
you can't use this. You have to use that.
Okay.
And the one on the rightmost side. Move really slow. Yeah.
Why is it so unclear?
Well, let me see. First, let's do too much light. Maybe a little bit too much light. Let's go back up. I'm just gonna pick a random spot. Okay. It looks like stars when it moves like that. Did you see that?
Mm-hmm.
Actually, this is pretty dense. So there's a lot of stuff in there. So we need to turn up the light a little bit so we can see into it. Let me see if we can find anything.
I'm gonna try to see.
Oh, look at that. She's not moving. Hold on.
I don't see anything.
Hold on. I'll get us there. Okay, now let's just wait. See, kind of move in there, in the center.
I do see something.
Yeah, let's just watch it.
Right.
Yeah, and watch this one. This one's going to be a little faster. This one's on the internet.
Which one?
Oh, no. They're the same speed. It's this one that's going to be a little bit behind. But yeah, we can see things moving and coming around.
A little behind.
Yeah, D-Money, this is a sample from a freshwater lake. And we basically just went to different spots in the lake and grabbed little samples and stuffed it in this jar here. Be really careful not to spill it because that would really suck. But I want to bring it over to the camera. Yeah, and see, can I see? Yeah. So it's kind of water sample, and we have a few other test tubes, a few other vials. This is actually yard water here. The one over here that I'm wiggling. This is yard water, or sorry, a soil from a yard with water added just to kind of separate things out a little bit. And then the other one's another lake sample. Ooh, hold on. I think we're really close to a big one. Let me see if I can't pull that down
A big moving stuff?
It looked like it. Maybe.
It looked like something over here. Like a line over here.
Let me get a little closer, a little better zoom. Okay, actually, let's crank it up to 10x and just see what it looks like. There's a little bit too much stuff in this slide, unfortunately. I'm gonna pick a whole new random spot. All right. Well, be right back. All right, I'm back. Oh, kids had to go to bed. Let's see what we got here. Yes, I just in the act of rearranging my my basement, I I was realizing like it's like, oh, I haven't played with my microscope in a while. And usually when I play with the microscope, of course, the kids play with the microscope too, and so I feel like it's good if I play with it from time to time, just to, show the kids, expose them to it. It's kind of fun anyway. See if you can find anything in here. Unfortunately, the lake sample, it didn't have a lot of plant life. So there wasn't a lot of algae. I really normally when I take samples like this, I like to get around the algae because there's always a lot of like plankton, little diatoms, and all kinds of other little single-celled or multicellular plant life. And with that comes a lot of other kinds of cell life too. So I'll probably, now that I got all this set up and working and have the layout working a little bit better, I'll probably do some more microscope sessions. But we were in a little bit of a hurry when that one was going on. So hold on a minute. I'm actually going to remake a slide with a little bit better of a layout. We kind of did that one pretty fast just because I wanted to make sure Chloe got a look before she went to bed. She's really wanting to play with this earlier, but we ended up getting sidetracked on other activities. So she's sad when she didn't get to play on the microscope after we spent, all day gathering samples and talking about it and getting everything set up. And so she played Minecraft though, so I think she'll be okay. All right, I'm gonna go straight to the yard sample actually. I was just really curious about that yard sample because I just went to a few spots where it was kind of muddy and gunky and got a few samples and hopefully didn't pick up anything too contagious or dangerous. All right, so let me do this facing the camera actually. Didn't think too much about the layout of this other than I just wanted to do this really badly. Oh, I hear somebody's footprints coming down. That must be Chloe. You left something? What'd you leave?
Oh, wait.
The cat pointer?
Oh, yeah, of course it's a cat pointer.
Good night, Lou. Uh-oh, where'd my slide cover go? There it goes. Nice and dirty. Should really clean these up. Actually, I'm going to do that. It goes a long way cleaning these things off. Get fingerprint oil on them and some light science before bed. Yeah, I'm always, me, I'm always doing something like this. I'm usually just doing it off camera. I never thought of doing this stuff on camera. My hope is like, people see it and they're like, oh, that's a pretty fun idea. Because I mean, when I was little, my teachers, I remember in high school, my teachers, what started with my grandpa getting me science kits and stuff when I was younger. But then in high school, my biology teacher, Miss Busby, my physics teacher, Mr. Miller, they hooked me up with all kinds of just equipment and books. And Miss Busby, in particular, she loaned me a microscope, and I could just use this high power microscope any day I wanted. And like, man, that changed my life. Like, could you imagine having access to that? And so I was, you know, I would basically go out and collect samples of water and from all over the place, soil samples, you name it, and bring them back. And I would draw them in a lot of detail. So I had a similar quality microscope as this one. I don't remember how expensive this one was. Let me see if I can find it. I think it was like $750 or something like that. It was maybe even 1,000 with all the attachments and stuff. I just like always wanted a microscope, and I was like, I'm just gonna splurge and get one that I love, and I've not regretted that. All right, let's as I said, I'm gonna turn down the light a little bit just so I don't like overheat. I don't know their sensitivity or anything like that. I am extreme amateur, but a little more optimistic about the yard sample. I mean, I'd normally be very optimistic about the lake sample. It's just I couldn't find like good spots for it. Let's see. What is that? Well, it's not an organism or anything. They'd be clearly moving, so get out of the way. There we go. And I'm on 4X, the lowest magnification right now. And per the theme, I guess I'll take another drink of my milk tea. He cha lo. Lo. Oh. Drink tea. This is a tea house. Nothing gonna move in here. Let's check out this little section. That's sad. My yard sample is just death. Oh, there's something. Little small as it be. Maybe. Let's go ahead and crank up the magnification, actually. Man, when I first bought this thing, though, I was exploring everything with it. If I could put it under a microscope, I was. Let's see. Man, these are tiny. So we're at 10x magnification right now, and I forgot what x this optic head adds. 0.5x? Oh, interesting. Let's see if we can crank this one way up to 40x. I don't have the oil, unfortunately. I fucking broke it. Sweet. Good job. And that's why I have 100 of these things. So the 40X one, it's you have to basically put the lens right up next to the slide, like, and it pushes in a little bit. But my slide isn't strong enough, and I probably overdid it with my strength. So no big deal though. We have, like I said, I have packs of slides on slides. We'll wipe down our slide and slide cover again. Well, sorry, we're wiping down our slide, and the slide is just that little glass piece right here that we actually apply the liquid to. And they have slides with concave lenses in the center, but I find that creates this sort of uneven depth, which allows certain things to swim and move freely, but also makes them harder to track, because just the act of them moving up and down in that water will bring them in and out of focus at a higher magnification. All right, we're going back to the pond water, or the lake water. I'm gonna stir it up a little bit, and we're gonna try to get a little less dirt than we did last time, but get that water. Get another little sample from somewhere else. All right, there's that slide cover. Here we are. Now nice and coated with my fingerprint as I accidentally smash my finger on top of it. Again, sorry I'm doing this off to the side, but that's just where my stuff is. Also, yet again, not a very good application. Let me see if I can make it visible. Can't really see it very well, but basically when you apply the water drop and you lay the slide cover on it, you want that water to go disperse all the way across smoothly. But sometimes if you get something that's thick caught in the microscope, maybe I can bring one of those things into focus. It basically doesn't allow the slide cover to go flush against the microscope or slide, and the water doesn't go all the way across. It creates a worse viewing. All right, should be coming into focus in just a second. We're resetting all the way back to 4x magnification, just because I always like to start at the lowest magnification that you can see faintly, vaguely the thing you want, and then you hunt it down, and then you find it, and then you can zoom in once you have it centered. Because if you have it centered, it's really hard to find things because the movement is so, so fast. But it'll appear really small here, and that's okay. So I'm just looking for any sort of movement. Yeah, this is some kind of boring water, actually. Another thing that's kind of fun to watch can be like take a kid's swimming pool, fill it with water. I do this at home. I just fill buckets up with water around the yard, and then I just kind of see what happens. And I'll go sample and look under the microscope. Let's see. There we go. Finally something. Oh, yuck. Yucky, yucky. Oh, it's a worm. All right, now we're going to click this in 10 real quick and see if we can maintain focus on it. Yes. All right, now it's focused. Or sorry, we got it centered. Now I'm going to try to focus it. Turn down the brightness a little bit, see if that helps. I'm going to try to do the coarse adjustment real quick and see if I can just get some better viewing in here. We're going to go back up. Look at that. All right, I'm going to play around with the fine-tune controlling now. Let's see if we can't see our little worm friend again. Just imagine when you're playing in that water, this stuff is everywhere, crawling all over you, in you. I'm just kidding. I'm just trying to cause some fear. That's pretty cool. Hopefully we can get a better viewing of it. I'm gonna turn down that light again too, because sometimes that light again it can kill them. The temperature. I don't know how sensitive all these things are. Well, I don't see the worm very well. Let's go back to 4x and get that back in focus one more time. All right, there we go, little wormy. And so this is a small multicellular organism. It looks like I'd like to see closer to see if it is in fact one thing, but at this size and that complexity, multicellular. Usually you see tons of like paramecium and little stentors. Actually, a stentor is like both nice. Both of these are on that image actually. This little green stentor over here. Let me configure OBS so it'll show my mouse. Browser image search properties. Show window. No, no, no. Thought there was some way to show my mouse. Oh well, we're not going to mess with that. So this little green looking on this window on the left, I'm going to center it or move it towards the top. It's called a stentor. This kind of little triangular looking thing. You'll see a lot of those, that Suctoria at the bottom, that kind of like these are all like really like you'll see these basically in almost any freshwater body you look at, or at least some variation of it. Like my last samples, we found amoebas, paramecium. My fish tank actually, my fish tank. Oh, have a good night, D-Money. Peace. Yeah, and so my fish tank, I wasn't properly, taking care of it, and I ended up growing a bunch of different kinds of algae and just singular cellular plant life. Uh, yeah, a lot like the Volvox, saw those. Spirogyria, Spirogyra, water silk. You can kind of see the little spiral filament inside it folded up or wrapped around. And little diatoms. There's basically kinds of algae. Anyway, let's get back to our feature production and see if we can't find some other stuff swimming around here. And turn up the light a little bit, see if that helps. Just looking for any kind of movement. Let's see, anything over here? Just at the edge. Sometimes you'll see there'll be things that kind of concentrate around the edges a lot. Oh, this water's just looking straight up dead. We'll keep growing the samples and I'll add random sources of energy and food and we'll see what we can grow. We'll get these things very very dense. We can make them as interesting as we want, at least until the population collapses or dies. But as I watch these samples I grow over time, they'll like subtly change in fun ways. Like the kinds of things that'll appear, they'll be really populated for a bit and then they'll like suddenly die off. And then maybe there'll be something left over, like a different kind of species takes over, or like you'll just become sparse with a few tough ones living. We actually found a tardigrade or two our last microscope scroll. Okay, I want to call this sample. I should have, I should navigate these a little more systematic, kind of just like row scanning it. And is my camera straight? Yeah, there we go. Now it's straight. What is that? Why is that all slanted? Okay, yeah, that's just not the edge of the slide, but the edge of our water sample. Because there's like a large surface that kind of stopped the water from evenly distributing across the top. Anyways, this was my yard soil plus water. I'm gonna get a better sample though. Oh no, this is the lake sample again. Yeah, it's the lake sample, but that's a kind of a boring lake sample. Again, though, when we were sampling this lake, one big point of difference was that it didn't have oh, it didn't have a lot of like algae or greenery, and so didn't get a lot of that in the sample. So this is a lot more kind of like lake dirt samples from the bottom of the shallow part of a lake, and had a few runoffs from creeks, and there's a spot where like ducks walked a lot, and I don't know, it looked like there's a bunch going on there, or could be, but But yeah, I'm gonna get some better samples. Today I just kind of wanted to make sure I can get my microscope up and going. Wanted to play with my kids as well because like we went and got these samples today, and uh, sometimes we find really cool stuff. There's a different lake by my house that we go to more frequently for samples, and as well as the ocean. There's some nice little creeks and runoffs around the ocean. All right, where are we at? Back at 4X. Well, dun, 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 dun. Oh, out of focus a little. Let me get that back in. Again, not. Crazy how life comes in all forms, and we're arrogant enough to believe Earth is the only planet with life. Claire, I 100% agree. And check this out. So, in my grand, journey to understand evolution and things like that, I've taken some fun classes over the years, like computational uh, genetics, like some kind of bioinformatics classes. And, we got to learn a lot about like tracking mutations over generations to like track life back to its origins or humans back to their, starting point, and experiments on E. coli as they've like observed it and how its DNA changes over hundreds of generations. And anyways, and of course you learn about DNA in the process, and that's made up of nucleotides and other atoms that all hold it all together. But the interesting thing is about these nucleotides, and a chain of 3 nucleotides. When I say nucleotide, I mean like the ATCG, the adenosine, thymine, cytosine, guanine, and yeah, if they're linked, 3 of those nucleotides linked together are called amino acids, and amino acids linked together in a way that they encode something, is known as a gene, and anyways, these nucleotides, they for a long time were like, how did life evolve on Earth? And we're like, oh, okay, so Earth had to both have sufficient environment such that it was able to form those molecules, the nucleotides, the basic components of RNA and DNA, and it had to be hospitable to allow life to evolve. And it was a big hole in like evolutionary study was like, okay, how did Earth make the first nucleotides? Like, what chemical process on Earth would be sufficient to create nucleotides? And we couldn't really do it. And the TLDR is that over time we started realizing that these nucleotides come from space. So we're being bombarded by them all the time. They come from meteors, and I think they're created from like supernova or some astral, uh, or like astronomical like phenomena. And the I can't recall what it is, but TLDR is they knew some of these amino acids came from space, but they recently have found all the different like primary amino acids responsible for life, and thus nucleotides in space. And so what that means is that life, a planet doesn't have to both be able to form the fundamental nucleic material, but it also and be able to be hospitable to life. It just has to be hospitable to life. And so if you look at the fact that nucleotides and amino acids are just being bombarded all over space, all over planets, to me that means the probability of life increasing on or existing on other planets, uh, you know, exponentially went up. It went up by some significant measure. I don't know how to quantify that exactly, but like if all of the base components of life, or at least RNA, DNA, are found in space and going hurtling through space and bombarding planet after planet after planet, I just have to believe there's definitely life on other planets. Also, when I do evolutionary simulations, like I program like little genetic algorithms in my code, uh, you know, uh, it's able to re-evolve a complex output, complex, simulation every single time. So when people talk about like, oh, the odds of evolution producing you is like winning the lottery 10 times in a row, it's like, yeah, if you go from absolute nothing or chaos into this highly ordered state in one step, that would be astronomically small. And but that's not what is happening. It's happening in iterations over time, and just that, strong survive, and thrive, and plus natural, or sorry, like basically natural selection combined with mutation, i.e., the fact that every single time a DNA or RNA molecule copy, there's a copy error. Uh, that's a mutation, and just that process and those two things are sufficient to create, basically a system such that it always produces that complex outcome. So I'm a little wordy, excuse me, but I'm going to find a couple cool experiments. Uh, Evolution of water organisms simulation. There's a really cool evolution program. Oh my God, there's a new one. I don't think I've seen this one. I'm going to go ahead and post it just in case it's cool. Uh, and whoops, and one more here. Carl Sims. Yes, this program here changed my life, man, when I learned about it. This next one. I'm going to pin that one. No, I won't pin it, but I do wonder if consciousness is arbitrary from all the life that exists. But then we're going into philosophical topics. Hey, and by the way, Claire, I love talking about these subjects. I have a lot of thoughts on like the boundaries of logic, and what's knowable and what's not knowable, and uh, the undefinability of consciousness. And sort of I don't know, I have some fun thoughts, or maybe they're fun, maybe they're crazy, I don't know. I'm just another human, but it's definitely my favorite subjects. Like all-time favorite subjects. It's sort of the motivation why I do anything at all. I don't know. I wake up every day in this universe, and granted, I'm pretty fortunate. I know that, especially now I'm out of poverty and living a much better life. But like, I'm just like constantly blown away the fact that we are here, the immense complexity of the universe, the immense amount of information in the universe, the fact that it's so persistent and consistent. You know, the universe remembers every little fucking detail about every single possible thing, I mean, everything, it's crazy. And so even not going, even not talking about philosophy, just thinking about the immense amount of information that the universe so easily, seemingly easily and instantly computes perfectly, like I'm amazed by that. I'm blown away. Like I'm not a religious person, but I'm definitely like I believe there's more than what we're able to perceive and understand. And so it's like I don't know, always kind of want to better myself and learn more and understand more. And so yeah, so I love science. I mean, I like everything. I don't know. I like art, I like science, I like reading, I like writing, even though I suck at it. I'm a bit of a robotic, mechanical, broken, choppy kind of person, just because I don't know. You can call it ADHD or mentally challenged. I don't know, but like, I just, I'm a little sloppy, but I have an immense interest in things. I have three theories. Either we end up in a state not unlike before we were born. We live our very same life over and over in an infinite loop. We wake up as another something in the universe. I think it's all of the above, man. Because like what I think is like, just go. Let's look at the most fundamental: nothing to something. How do you go from absolute nothing, the unspeakable nothing, to something? Like, how do you go from absolute nothing to a square or a circle? You can't. Like, there's nothing to go from. You can't say I randomly go from nothingness to a point or a square or there's like randomness is a something, and so this is called the nothing to something problem, and I think it's a logically unanswerable. Like, I don't think you can go from nothing to one something and not something else in the process. And what I mean by that is like there's just not a path from absolute nothing to a something. And you can't just hand wave it away and be like, oh, it just quantum randomness resulted in a something appearing here. It's like, no, then we're not talking about the fundamental thing, because what is that randomness? What is that RNG? And so I subscribe to a more simple philosophy. I think it's like in the same way, if you look at the Boolean word Boolean, and there's the word, Boolean is defined by true or false. And so if something's Boolean, it is true or false. And you don't define true and then define false, and you don't define false and then define true. They mutually define each other. It's like it's either true or false. Like, and so that mutual requirement, that kind of duality between these two terms is like, I think we're onto something here. And so in a similar manner that you can't have true without false or false without true, I kind of think about it like the universe. Let's just assume it should be nothing, right? Like, why in the hell why should there be anything at all? Why is there anything at all? And so let's start with that. Why isn't it nothing? And it's like, well, what is nothing? What is nothing? Absolute true, the unspeakable nothing. It's not, nothing is not a square. It's not a triangle. It's not a point. It's not a you. It's not a me. It's not a this universe. It's not a that. And what I'm getting at is, I think in the universe, I want to personify the universe a bit, but in the universe's sort of attempt to be absolute nothing, it's like, it can't logically be absolute nothing, because to be absolute nothing implies this concept of what is not absolute nothing, to give that meaning. And otherwise it has like no meaning, kind of like the true or false. You don't have the meaning without the other. And so I think like almost like in a big bang, absolute infinite big bang, it covers everything. Like the universe, again, personifying it, tried to be absolute nothing, but in that, paradoxically, it's required to be, you have to know the what not nothing is. So nothingness, true begets false, and nothingness begets everything, like not nothing. It's like true and false, or let's call it true and not true, maybe a simpler term, true and just not true, instead of inventing a new term, false, true and not true. And similarly, I think it's like nothing and not nothing. So nothing requires the existence of not nothing for it to have meaning. And I think that's kind of like when you look at like the heart of like, like the Zen or Eastern philosophies, or hell, even Christianity, like they talk about like the absolute infinite God. And I think like the universe is absolutely infinite. I think it's absolutely infinite. Paradoxically so. Like beyond description, like time exists and doesn't exist. Kind of like how in your mind you have your current idea of time and things that are happening now, but all your other life memories are still stored in there and can be activated, recombined into a thought at any time. I kind of imagine the universe is like every single state exists, or potential state exists, and like we're navigating through it as a layer of it. You're navigating through a layer of it. There's something like higher up experiencing multiple layers of it at the same time, and then like at the apex is like the absolute, the realization of absolute everything, like heaven, hell, yin and yang of like every single concept and possibility. And so I don't know, like to me it is the only thing that had like a logical, like simple answer to it, because nothing to something problem I find intractable. And nothing is possible to imagine, I think. Yeah, exactly. I mean, I'm going to argue further, nothing is impossible as a concept without having the definition of what that is. Like, I think like the universe paradoxically was not able to be nothing, because nothingness doesn't make sense unless you have the inverse, not nothingness. And so To me, like that was like that realization is like it wasn't satisfying in the way that I was like, oh yeah, okay, boy, I understand everything. But like it, I think it's an easier concept to grapple, and the idea that the universe is absolute infinite. I mean, I don't know. I know it's a bold statement, but it just seems to be the simplest kind of way to look at it. And so uh, it's a little scary, though. The ramifications of absolute infinity to me are almost like I almost get like a panic attack thinking about the immensity of that, honestly, and what that really means. Like think about like the Boltzmann brain, or just this uh, the magnitude of what it means, the ramifications of an absolute infinite means. It means there's a you doing every evil thing. I mean, like it's it's pretty messed up and beautiful and scary, and it's just well, it's just everything at all. And they say with the way time works, everything that has happened and will happen has already happened simultaneously. We're just forced to experience those moments in sequence. I think so, man. I think that too. Like I think even like free will. If free will exists, I don't think it's definable. What does what does it mean to like I control my own thoughts? Do you really? You control how a physics a rule operates? You control the charge of an electron and proton and how fundamental physics work? I just don't think so. We're along for the ride. And personally, as long as there's some way for my will to persist in some form, I'll fully accept whatever the universe is. I totally agree, Claire. So that's actually something like people talk about like AI too, and right now it's clearly not conscious. It seems, at least it doesn't seem to be. Uh, but like if we could ever get to that point where we discovered another way, like other ways for consciousness to persist, I think that'd be great because then we could travel the stars and I wouldn't feel as bad if there was an AI takeover. We're talking like, the future real AI when it's like embedded in the chips and there's neural network chips directly printed and, it's generation thousand of this shit and, whatever that future looks like, which sounds scary to me. It sounds cold and bleak. As a human traveling around through space trying to survive because Earth is uninhabitable, honestly seems pretty dark and scary to me. And so, yeah, I could imagine like there's better forms of life that are more suitable to explore the universe. I don't know. I'm curious how it'll evolve. I hope humans tough it out. I think we'll have to like genetically alter ourselves, if we want to survive in the future. But I'm talking like, millions of years or whatever that looks like. At some point, we have to skedaddle and get out of here if we want to live. Maybe being a little optimistic will last that long the way things are going these days. I'm amazed we made it this far. But I don't know, it's a fun little journey. All right, this is the most boring water sample I've ever taken. I am definitely going to be back with a better water sample. But yeah, ever since that like nothing not nothing kind of paradox realization I had. I was like, oh, I assume cause and effect. Like when you assume nothing to something, you're sort of assuming cause and effect, right? A to B. And I was like, why do we have cause effect as such a fundamental part of our logic? I was like, oh, it kind of makes sense because, a neuron is either sending a signal or not sending a signal to another neuron. A neuron either fires or doesn't fire to another neuron or set of neurons. And so that's kind of very cause effect at its foundation if you think about it. And so it's like, oh, that makes sense why so much of our logic is cause effect and a if A then B else C, that sort of like Boolean branching logic. And so that also helped me understand how to like all human logic can kind of be tracked back to that sort of serialization. I have a concept for this scenario. I have no clue how to write it in short story form, but the concept is a forever machine with two human minds that exist after the universe reaches full entropy. The concept involves the two brains communicating with one another to keep one another company. I'm glad you're considering that because I would hate to be alone for millions of years or whatever it is. I think of like Fallout 4, the brain in the jar. I'm like, ooh, that would be terrible. and see if they can prevent each one another from reaching madness. They cannot die, and so are forced to have to persist. Man, yeah, it'd really suck if your like partner brain had a mental breakdown though. And you'd be like, no, man, please stay stable, stay stable. All right, I think we got this one. Let me fine tune adjust this one. Maybe the other way. There we go. Finally, something worth viewing. Again, I owe us some much better water samples. I'll preview them before. I'll probably just start feeding these and I'll nurture a few samples for us. Yeah, yeah, great. We're looking for Dragon. Is this Dragon? This looks like a Dragon. Look at baby Dragon. This is before Dragon evolved. So if we can destroy this cell right now, this will prevent Dragon from ever evolving. This is the cell that I deemed to be responsible for Dragon's evolution. Look at others eating stuff and making poop, and let's just watch this. We finally found some action. I'm not sure. I don't have a good look at it, unfortunately. Uh, I'm pretty familiar with, maybe like 10 or 15 different things, but I also can't get a good look at it because it's a view. I would go heavier, but let me look around. Oh, I have a few photos I downloaded in preparation for this. Oh, let me get it back in view for us. That's most important. But I want to get some water with a lot more plant life. The plant life, I feel like the micro, like diatoms and such, like I feel like they always have a lot of that kind of water has a lot of stuff going on in it. Uh-oh, Operation Get Unblurry. Everything's like, oh no, we're going the wrong way. Uh-oh, our suspect got away. Baby Dragon on the run. Oh shit, it's gonna mutate. It's about to mutate. Oh, there it is, Baby Dragon back in Spectrum. Thanks for keeping us on topic, Grape, on Metroid. I almost forgot. I forgot this is a Super Metroid stream. Funny how even the most microscopic of creatures have to eat, absorb nutrients, and poop. It is, man. I want to find some. There are some like really cool ones. I'll get some. Like there's some front feeders that they're kind of like triangular shaped with a ovular shaped mouth, and they'll have cilia around the top, and they'll just be like spinning them around, and it'll pull the water in like a current, and they'll just kind of like filter feed, and so you're gonna see a lot of different types of life, and you can kind of, I don't know, you can almost imagine like, oh wow, that category exists of life. It's kind of like how when you look at viruses and you're like, oh well, that's first the first fundamental parasite. uh, oh shoot, Gameaholic finishing his seed. It's playing a hard tricky, I think. That's why I get paid the big bucks. Heck yeah. Yeah, I was just saying, Grape. This is not the most interesting water sample, unfortunately. All right, we're gonna recycle it. We're gonna do a quick scan. If we don't see anything in the next minute or two, we are gonna swap out our water sample. I always take the water sample, by the way, and I drop it back into the original container. So I take fresh water in my or used water, and then I just run it down the slide and let it drip back into the slide, and then I wipe down what's left. Like this is already pretty damaging to a lot of the life. Like they die after this, but I really don't like killing them any faster than I already am. So I try to just kind of drop them back into their original environment. Worst case scenario, there's a bunch of nutrients in here for the future ones. All right, slide change. I want action. Dun, 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 dun. Dun, 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 dun. Boom, 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 boom. Great. Both of your runs today, they were what, 40? Was it 48? I can't remember if it was 48 or. All right, we're gonna crazy with this sample. I don't know what to expect with this sample, but we're gonna go excessive. We're gonna put a lot of extra stuff on it. Darn it. Always clean your slide cover before you start putting your stuff on your slide, because then you can just put your slide cover on top of your slide directly without interruption. Also note, if you have two slides stuck together like I did, separate them. You'll get better result with one slide than two. Today's common sense lesson. All right. So dropping the slide cover on. Yeah, I give that one a. Oh, now, okay, now this is good. This is what I was looking for. You want the water to kind of, I don't know if you can see that really well. You can't see it too well, but you want the water to sort of, like when you put the slide cover on top of the water drops, you want the water to just expand all the way and evenly out all the way across the slide cover, so that, there's no air bubbles, and it goes all the way to the side, and it's roughly the same depth all the way across. And I'm gonna take a drink of our milk tea here, since this is microscopes and milkshakes, or milk teas. By the way, I'm like a bad Monte Carlo tree, in the sense that, like, I always missay words. It's just like, and then after I say it, I'm like, oh, I didn't mean to say that. I'm definitely a talk before I think kind of person, stream of consciousness. So yeah, if I say stupid shit, just like, no, it can't be helped. Mm-mm-mm. Let's see. All right, let's check out our new sample here. I shared another one of my step charts in Discord. Heck yeah, I want to play it. I got that set up on my computer. I forgot the name of it already, but I have it installed. If you wanted to say the name of the game in chat, that'd be sweet, so I can remember it again. I used to play, I don't remember what it's called. I played one of those where you use your keyboard a long time ago. Never got super good at it, but loved it. Mostly because it looks really hard. Let's see. Are we just going to get stood up? Nothing happening. The best water I ever viewed, though, was my fish tank. Because I had a fish tank, and, of course, I got the fish tank, and everybody in the house was like, yeah, we'll help you out with it. Oh, yeah, we'll help you. We'll help you change the water and all that stuff. Nope, nobody did anything. It was all me. And we had 16 fish. It was my first time ever raising a fish tank with that many fish, and those things shit so much. It was ridiculous. It was like unbelievable how much they ate and pooped. And so we ended up having to clean it like every week. And it was so much work, man. So much work. And, I'm sitting here like measuring the acidity and blah, 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 blah. And finally, I'm just like, man, this is crazy. I'm just going to, the fish started to die too, like one after another. And as they started to die, the fish tank simultaneously became more maintainable. It's like amazing when you only have 7 fish left, how much easier it is to keep it cleaned. But eventually that became a nuisance too. And I just wasn't ready for all the work involved, and I'm busy. And then, all right, we're going to go back up to 4X just to see if we can find anything. And we'll zoom in when we find motion. But anyway, so I just started letting algae grow, and because I saw the algae growing, and I had some snails and stuff that I brought in from the water, from the ocean, and it ended up growing like crazy. but it formed a sort of equilibrium in my aquarium. And anyway, TLDR, months later, like a few fish ended up living for a long time. And the water, like I didn't even have to like really clean it. It just like there's an equilibrium. And I mean, it was a slowly decreasing, it was slowly, going towards chaos. I knew the good phase was going to end, but it was cool how long it lasted without me having to clean it. Like we're talking like 6 months, like, and it produced all kinds of life. And then even after the fish died, I kept the aquarium around and just kept, like, I'd put different things like sugar in it or something, or I'd go scoop up water samples from outside and just drop a few teaspoons in. And then I would just sample it over the months and look at it on the microscope. All kinds of stuff would grow in it, and it changed colors and probably wasn't healthy or safe, but man, it sure looked cool. But unfortunately, like I just, I wasn't streaming or anything, so I couldn't really show it off. But that will change. I'm going to rebuild my little sample. And once I find some cool stuff, I'll share. Okay, we're going to chalk this up as a boring sample too. We're going to start recycling our samples a little faster just so we can, try to find something cool before the night's over. I just want to find us some really cool stuff to see for the night, and then I'm going to call it. And I'm going to be working. We're doing a hackathon for Tuesday, Wednesday, and Thursday with the team, which I'm looking forward to. But I'm going to be hanging out at the Airbnbs with them, and so we'll be a little bit. I won't be as available. All right, here's another sample of lake water. We're going to see if we have some better luck with this one. Down. Oops. Oh boy, this sample looks interesting. Hope it's as interesting as it seems. Uh-oh, dropper. Okay. Find my slide cover. Just gonna kind of drop it back on top. All right, we got a pretty decent spread on this one. But again, I fear we're not going to have a lot to look at. Oh my God, we already got something. We already have something, guys. Oh yeah. Go to the left. We're going to turn down the light as to not aggravate it too much. What is going on here? We're going to go to 10x and do some fine tuning. All right, we're going to come down a little bit. Yeah, look at that. Look at that. Don't move, guy. Don't be shy. There we go. We finally got something. Unfortunately, it's a little thick here because there's just a lot of material. This is what happens if you add too much water or too much material. It's kind of an art. Mm. I love this microscope. I highly recommend it. I'll drop the link here to it as well. Like me getting a microscope when I was in high school from my teachers and my grandpa when I was younger, just like, oh, it just changed my life. Unable to pen. The fuck, why can't I pin this? Like, I just can't pin links on. Let me see if I can pin it. Let me see if I can pin it in my. You know what? Who cares? Moving on. We are here to look for organisms. It's like Pokemon, but microscopic. Dude, that thing is doing some nomming, man. Look at that. It's going wild. See if we can get a better view of it. Ooh. All right, we'll move on. All right, we're gonna go back to 4X. So we're gonna zoom back out. We're gonna see if we can find some general movement and then zoom back in like before. Get some focus. Again, it's way easier to find things in a lower zoom level just because, like, I mean, I think it's pretty obvious, right? Like, if it's moving, it moves too fast when you're zoomed in. What is that? What? Oh, I thought it moved, but it did not move. We're still going to zoom in. I'm just curious. So I'm going to get it right here in the center, and then I'm going to go from 4x to 10x, like so. I'll crank up the light a little bit, see if that helps. Nope. I'm going to zoom in and out and see. Oh, okay. Definitely some filament structures. They're not moving though, so that's kind of interesting. Wait, is my stream? My stream is working, right? Okay, phew, wasn't showing up. Okay, phew. Dun, 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 dun. That's pretty cool. All right, next spot. Let's keep moving at this zoom level. We'll see what happens. And so you kind of zoom up and down because you're actually seeing like different layers of the water sample because it's deep enough that the focal points will actually, yeah, there's multiple focal points within the water sample. So yeah, this is basically a lot of sediment. They're just like small rocks and sand from the bottom of the lake. So I think I messed up when I got that. I should have got a little bit more vegetation. There just really wasn't a lot on the side we were in. And then it started raining and we had to leave. And so I was like, oh no, I just waited too long to get the water samples. So definitely like, it'd be more interesting to look at your saliva maybe or your blood. We can see some blood cells and I don't know. I've looked at about everything you can look at. Got something wiggling around. There we're gonna go. There it is. Look at that. Oh, we can see it. Another. Dude, that thing is big. Look how long this thing is. Oh my god. Two of them. I'm gonna turn down the light again because we don't want to distract them. See if we can get a good view with this level of light. Lower the light, the better, because they'll react to the light if they're sensitive to it, and they'll like swim away. So like just the act of observing them causes issue. Well, I might have to turn up a little bit more because I'm losing them and not able to focus anymore. No. All right, back up to 4x. Let's find them again. We lost them. Zoom out. There they are. Okay. Get a little bit of zoom back in, or focus. And what I'll do is I'll look at some of these offline and I'll try to classify them for us. I was going to do that live, but I decided it'd just take too long, and I don't want to like sit here with a sample on under this light for too long and cause damage to the samples. All right, I'm just trying to turn down that light so we can see it. There we go. There we go. Let's just watch it. Wow, it's really long. You can actually see the material moving through its inside organelle, like its digestive organelle. I don't know if it's called a vacuole or what you would call that. Honestly, I'm not sure of the organism, but like you can see the material in it at least. Looks like a little microscopic worm. Let's actually we have this image search pulled up. Let's see what all it has here. Amoebas, parameciums. It's definitely none of those. Stentor, didiniums, suctorias. None of those. Large free-living ciliates. The stentor. Contractile, collect food by cilia at end of mouth. That's what I was talking about earlier. Under the phylum Ciliophora, order Heterotrachea. Oh my God, I remember Miss Busby's biology class from high school, and we covered a lot of this. So this is kind of, yeah, let's see. Lacrimaria, ciliates with long necks. But yeah, note the distinctly big body and small neck. This is not that. Microscopic organisms, the rotifer, these are pretty common. 100 to 500 micrometers, so like almost a half a millimeter. That's pretty large. And let's see. Hydra. And the first time I found a Hydra, I was like, whoa, what did I discover? And then I discovered a few days later, I saw freshwater jellyfish. And yeah, planarian. So the first when I first saw this, I was like, is this some sort of planarian? But I didn't see the eye spots. And so this is what I was thinking, like a nematode. Like these are just little basically roundworms. But I don't know. Not confident enough to say. Tardigrade. These are really cool if we can find tardigrades. Earthworm. Oligochaeta. Yeah, not. Wait a minute. Few millimeters to 1 centimeter. Maybe it's segmented worm motion hair bundles. I mean, it looks kind of like that. I'm not confident enough to make that decision. But yeah, like I've seen all of these Daphnias, copepods, ostracods, amphipods, mosquito larvae. Yeah, those aren't really microscopic, but there's, yeah, exactly, 0.2 millimeters. But I've seen a lot of Daphnia. Okay, let's keep some of these up. These are promising. And let's go back to our hunt for more organisms. Yeah, and Claire, you're mentioning the diversity of life. Like, it's immense when you think about the diversity of life, right? Like, I mean, look at the insect world, and just imagine if they were on a planet where they had a different respiratory mechanism that allowed them to get larger. Because a lot of insects are limited on their growth because they breathe oxygen through their body, so it's a surface area to volume problem. They don't have the efficient respiratory system and lungs like we do, and so they can only get to a certain size, which is also why like prehistoric insects were much larger, is because the oxygen content in the atmosphere was larger and allowed them to get larger because they could get more oxygen. And anyways, like, so when you look at things like aliens or hell divers, and like you look at the crazy, or books you read where there's like crazy animals and aliens, like, I don't know, it's not that insurmountable to me. Yeah, man. Yeah, it's a solid microscope. I I invested into this thing a few years ago, and I really love this microscope. All right, we're going to try to go back in. We're going to go to 10x here. Let's see if we can do it. So we got it centered. All right, let's quickly try to get it into focus before it moves away. Oh, yeah. And I have you can see this little adapter on the top grape. So it like connects to my computer. Oh, there we go. Oh, delicious. I mean, not that thing doesn't look delicious, but this view is fucking good. Was good. Get back over here. Get over here. Yeah, that definitely looks like a some sort of worm. Alligator. We need some microbiologists on here. Don't miss the smell. Yeah, I actually wish we had a microbiology class. Like I didn't get to take one, but needless to say, I still spent thousands of hours playing on my microscope growing up. I love my microscope. I had, I started off with like this simple little children's microscope that my grandpa bought me, and just then my teacher, I don't know if you were on when I mentioned this, but my high school, so I grew up all over the place, man. I grew up, we're moving 1 to 2 times a year, total chaos. I've lived in towns of like 100 people, but I moved to this town called Pocahontas, Arkansas, up in northeast Arkansas, a little old town, a few thousand people at the time, 5,000 people. But it was like a bigger city I'd lived in. But the surprising thing was, despite that relatively small size of a city, like we had a really good school and like good teachers. Uh, like we had a teacher who previously worked at NASA, who was our physics and calculus teacher, and he got me into physics and spent a lot of time mentoring me and stuff. And our biology teacher, Miss Busby, and she was very nice and very just like made biology exciting and fun. Oh wait, is my screen frozen? Looks like it's not updated on the newest image. Hold on a minute. Let me see here. Microscope. The capture froze. Oh man, I hope the capture wasn't froze too long. There should be a big worm here that everybody should be seeing. Major bummer if that was all if all that was like frozen earlier. That sucks. I was having problems with OBS in its window view. It wasn't able to see this application actually. Yeah, I'm so sorry, man. We've been having some like spectacular views over here, and sort of these basic little worm structures. Yeah, yell at me if it freezes again, somebody. Oh, we'll go down actually. We'll go up. Look at that. Much better. Oh man. So as long as I look at my OBS screen, which is reacting about the same time as my real app, is that. So as long as I'm looking at the OBS screen, I'll notice if it freezes from now on. So this is just some sort of microscopic worm. Multi-celled organism though, not single-celled. Yeah, so the difference though, Grape, is that if I use the legacy window source, it might freeze, but it'll come back eventually. If I use the Mac OS video source, like there's like a Mac OS window source, or what's it called? Mac OS screen capture, it will freeze and I'll have to manually restart capture to get it back on. And so normally I use the legacy window because the screen will always restart without me having to restart the capture. But for some reason, it couldn't find this window of this app I'm using. It was like driving me crazy. And so the only way I was able to do it was to share the whole screen and use the Mac OS screen capture source. And yeah, it's a mess. Oh, dude, I have terabytes of free space, and I'm running a pretty powerful machine. Yeah, this in this era, I've heard that issue before, but I don't think it I don't think it applies to my computer with the amount of like the power that this thing has, and it could still, of course. I don't fully understand the exact nature of the bug to a technical enough degree to declare one way or the other, but I've been having this issue on this computer since early days. There we go. Look at that head just peeking around. He's probably like, oh, this water's getting warm over here. Got too full. Yeah, I was hoping it was something like that, but I have like 1.2 terabytes or 1.6 terabytes left. Again, not like infinite space, but should have more than enough space for buffering some video. All right, we're going to zoom back out. We've been watching those worms for a while. Let's go see what some other worms are doing. I'm sure those worms are like, can you please let us eat in peace? We're just going to zoom to a random position. This is it right here. Dun, dun. Zoom in. Again, this is not, unfortunately, the most interesting water sample. I usually, if I grab water, again, I try to get like water with algae and things like that, and there's just all kinds of life. It's basically like a salad buffet for a lot of the organisms too. So it just creates a nice, diverse, healthy, interesting, fun to magnify life. All right, we're gonna go zoom in 10x and let's find. Oh, geez, there it is. All right, let's do some fine tune course control and get this in focus, or screw it up and get it out of focus. I'm gonna adjust the course control real quick. There's a little air bubble right there. You can see its digestive tract as food is going into it from it eating. Yeah, and you can kind of see some of the segmentation on it. So I think this is just that, uh, you know, I'm not confident to say it's this literal oligata or whatever, however you pronounce that, but oligata, but it's some sort of microscopic worm I'm going to go with. Uh, oh, that's pretty dense. This sample is dense with worms. This is awesome. These things, all their life is just constantly moving around, nonstop eating. And you'll watch them eat each other, and you'll see like an amoeba come along and just absorb stuff into it, and like you'll see like paramecium just there, and then 1 minute like something's exploded it and it's leaking its guts around. Like I've just seen all kinds of crazy stuff under the microscope. And I'm the kind of person who walks barefoot everywhere, and I don't really wear shirts. I only wear shirts on stream because like I'm trying to be somewhat professional, but I hate shirts, and I don't know. I like being free. And so after playing with microscopes, like I'm like a little bit like, oh shoot, do I want to walk in my yard without shoes? I do it anyways, but like I think I think back to a lot of these crazy things I've seen, and I've seen some freaky looking things over the years. I used to have a whole deck of hundreds of index cards. All right, I'm going to zoom back out and find another section. But I used to have like hundreds of index cards from just different organisms. I would draw because I'd find it, and then I would draw them painstakingly because sometimes they move, and then I would go look it up in a book and try to classify it. Mm-mm-mm. Let's see. Just choosing kind of random spots again. Jump, 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 jump. Also, there's not a lot of coloration here too. Sometimes some of my samples will be like very vibrant in colors, akin to a coral reef, except microscopic. A microscopic coral reef without coral, and really without being a reef. It's just colorful. Let's see. All right, we found you. Zooming in. We're going in. Ooh, 40x. I'm sorry, 10x. Oh, almost perfect. Right on its. Go other way. The controls are always inverted for me. These things seem to be just moving around pretty much unchallenged. Wow, this is a really long one. Ugh. Imagine it crawling up your toenail and getting embedded in there. And the only way out is to eat into your toenail, the space between your toenail and toe. I want you to think about that every time you walk in or swim in fresh water outside. But no, like if when you take samples, all jokes aside, when you take samples from like ponds and like really still water, small bodies of still water, you find a lot of funky stuff in them. Like I don't swim in ponds anymore. I used to swim in ponds. Like I had no limits as a kid. I was like, man, we just had no limits. We're little country, little country hillbillies, and and I say that with sincereness. I love my Arkansas roots, in the country, and so oh, what was I gonna say? I don't remember. Anyway, it was fun because oh yeah, the ponds. So again, my teachers loaned me microscopes. We had access to all these different water, like bodies of water, and we just find all kinds of stuff. The little dot pattern is interesting around these. It's pretty consistent between all the worms we've seen here. Hey, what's up, Mr. Fox? Oh my word, we have got some stuff to talk about when I get done here in a second. This is the new version of Multi-World, Mr. Fox. This is Super Metroid Multi-World where Samus shrinks down to microscopic organism size and probably just gets eaten by these things because there's a lot of them and they're very ferocious. They just eat nonstop, which might not be a bad life if they have any sort of pleasure. If it, if I don't know, it's probably just pure input output without any of that though. I don't know what size or point in evolution where things like neurotransmitter responses like dopamine and stuff come into play. Maybe it is at this level. I doubt it though, but I'm a little ignorant to that. I won't pretend to have deep knowledge when it comes to microorganisms. But I do love exploring and watching them. And then when you just think of, this thing has DNA in it that encodes all of its organelles and structures, much like we do. You think about how we might have like 60% DNA in common with like a common fruit fly. Sorry, grapes on here, and I'm thinking about grapes when I say fruits. Uh, And trees, we might share anywhere 25 to 50% with certain trees, and I just think that's pretty cool. It'd be cool if we could find like a tardigrade or something. What is it eating? Try to focus in on the thing it's eating a little bit. Wow, you can definitely see the worminess to it too. Like just from a biological perspective, like what an effective design, easily modifiable, easily adaptable to swimming and water, land motion. Pretty simple structure in terms of like, here's how we encode a digestive tract that runs through this long cylindrical thing. Just seems like a stable species structure, which I guess is why you probably see that pattern over and over in biology. Dude, it'd be badass if a microbiologist came on our stream and was just like, "Oh yes, that is the Alogaceta of the phylum Annelida, class Oligaceta. Yeah, yeah, they're very common. Watch out, it has a microvenom in it that can kill a human, and they like to embed into human eyeballs if you swim with them." That's the information you're gonna get from me if I tell you what they do. Let's see. This thing just doesn't stop. Oh, we can see a really clear view of its digestive tract. Let's see if we can watch it and actually track food going through it. Like these things, things process quickly at this scale of life. Like there's some organisms you can just straight up watch the food digest. All right, we are going to swap our slide sample. Let's say goodbye to our friends here. Oh man, I'm gonna be very careful to use water and uh, basically I want to drop water on the slide and have all these go back into the its container environment. Like I want to try not to kill them. I'm not going to crush them. I'm not going to throw them away. Maybe that sounds silly. Like I've pretty much already doomed them to die by capturing them, but like I still try to handle them with respect. Like that thing has no idea what's going on. He's just like nom 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 nom. That's literally all these things are doing. Just 24/7. I don't even know if this thing sleeps. I'm gonna guess it just does this 24/7 until it dies. Oh, getting out of view a little bit. Wow. All right. Pull and slide. Say goodbye to our micro worm friends. And again, I don't think those are nematodes. I think those are like, oh, I don't know, it might be a nematode. So there's that nematode up here, but then there's this one down here. So there's a nematode at the top here. You can't really see my mouse. It's the image immediately below my profile or my webcam, or 1 to 10 millimeters, few millimeters. Yeah, I'm not really sure, but I think we were looking at these. Ugh, a few millimeters to 1 centimeter. Worm motion, hair bundles, and segmented. We saw the segmentation, and we also noted those kind of red looking dots that you see in the image. Ours was very much covered in that. So I think I'm going to feel 85.32% confident to say that that is probably what we're looking at there. All right, we're dropping our guys off. I'm sitting here saying I was gentle, but like the act of the slide moving was probably the equivalent of like a magnitude 9 earthquake, and I probably killed them. This is really hard to do too because my water dropper has a leak in it. Great. I was getting excessive. It had a rock stuck in it today, and I was like, if I squeeze my dropper hard enough, the rock will shoot out, like a kidney stone. And then all I did was I broke the dropper. So I think there's a lesson in there to not force things. Uh-oh, where's the slide cover? Yep. All right, we got our slide. Our slide is clean again. I'm gonna try not to fingerprint it. Our slide cover is clean again. And we're just reusing the same one. I have stacks of slides and stacks of slide covers, but don't feel like. Yeah, okay, so I'm gonna do it again. We're gonna go deep. We're gonna go deep. It's a deep sample. Just imagine these things are trying to swim away and they're like, we're gonna go down. And I'm like, good luck, bro. Okay, we're gonna get samples from multiple places. That's what we're gonna do. We're gonna create a zoo of choice. A zoo of a sample. All right. Dude, I even mixed 2 different test tubes together. It doesn't matter though. They're from the same lake. There we go. Oh, much better. I improved my technique. At first, all the dirt clumped together, and then I like lifted it up and moved it a little bit, and everything resettled. Okay, so right now we're in 10x magnification. We'll see if we see anything. Otherwise, I'm going to jump right back up to 4x and start from the top. We'll see if we can't get lucky with a few random motions. Oh, let me get back on the slide. All right, there we go. Oh, we got some. Hiding out under here. Always like moving in the wrong direction. Mm-mm-mm. Haven't had a milk tea in a while, so this is nice. Okay. We're going to back up to 4x and see what we can't find. I'm going to turn down the light a little bit. Yeah, the light's good. It's not too high. Again, I'm trying not to boil them. Not literally boil them, but they don't like the light. What are you munching on? Oh, more worms. I think we just have a sample of a lot of worms. We more or less collected a beach sample, which explains a lot. So you can just think when you're walking on a beach, there's these little microscopic worms everywhere. Yeah, and thanks for everyone who's checking this out. I'm glad you find microscopic things interesting. I find this level of life to be really cool. Again, this is definitely one of the weaker samples that I've observed. So I want to make sure that we get a proper microscope exploration. So I will take that as my responsibility in addition to KPDR sub-hour runs. and get it done. Let's see. Go up. Oh, no, it went down instead. All right, I'm going to go right. God dang it, it went left instead. I swear it's just inverted, and I never get used to it. All right, let's go in deep to 10x again, see if we can't see anything. More of these worms. That is all we're really seeing. Another one. I want to crank up the live view real quick and just see what happens. Hold on. It might slow things down. Let's see if there's like better quality. Yeah, I didn't realize that whole time we were producing like a 300 by 300 something pixel image. Now we're producing a 1792 by 1374 pixel image, so it should be higher quality. More, oh, well, we learned a little bit more today. Next time we'll start off with higher quality images out the gate. It does affect scroll speed, but it doesn't seem to be affecting it too much on today for some reason. Oh, we're in a land of darkness. This is the center of the slide where there's a lot of soot and soil kind of consolidated. Let's zoom out and see if we can't find something interesting. We'll crank up the light a little bit so get some nice contrast. Oh, there's something. I bet it's another one of those worms, isn't it? Yep. Oh, don't wanna roast it. I like how it scrolls like that. It looks like you're traveling through space. Warp 1, warp 2, warp 3, we're gonna go warp 10. Bam! We warped across this slide sample into a whole new quadrant of space. It's like a roller coaster for the microorganisms because they feel that acceleration. It creates like a little wave. Can't see it very well, but you can make the water wave. I don't really know if they like that. Yeah, this is just a barren wasteland of worms. Look at that little shell. You can kind of see. Ooh, we're gonna explore that. I don't know if that's something. Yeah, you see that? Could be like a dead multi-celled organism. We're lucky. Oh yeah, and look at the hairs. It might be like a. Ooh, yeah, you can see the little sections of hair coming out. Since it's dead and not moving, we're going to be able to zoom in on this one even more. If I don't break my slide again, I accidentally, total noob mistake, but I broke the slide cover because I zoomed in too much and it just wasn't, the slide cover wasn't as strong as I thought it was. So this looks like just a body part. I think this is just a body part of an organism. The cool thing about this is you'll see like bacteria and smaller single cell organisms consuming on it. Basically, if there's something dead, there's some microorganism slowly eating it. So I thought I saw one too when I said that. Let's look around and see if we can't zoom in and find vulture, some vulture organisms. I mean, kind of looks like a crab leg if you look at it really abstractly. And so these little organisms probably think they found the world's largest crab leg, and that's how happy they are. All right, we're going to go in deep. This is a significant 4x increasement, and I don't know for sure if we're going to be able to do this. So this is going to be a little bit of an experiment. Is it? Oh shit! I don't think we're going to be able to. All right. Slowly. So we have the slide pushed into the lens essentially. That's how close it is. This is not an oil level high magnification, but I'm having issues. I think because the slide water and stuff is just too thick for this high level magnification. But yeah, let's try. Let me get in there. First, let me check and make sure the lens has enough, like kind of. Yeah, okay. Let me take a Q-tip. I want to clean it out real quick and just kind of make sure the lens is clean. I'll do that with all of them actually. I haven't really. Yeah, the 100X is an oil lens. I don't have that unfortunately. I found it. No, I do have that oil. I forgot. I found it in the garage today. Okay, let's try this one more time. The 40X. And again, I'm going to get it a little close. Yeah, I don't think this slide can handle it. For that, I need some stronger slide covers to give me a little more confidence on this. So we're gonna not do that. I'm hearing it crunch and crack a little bit, like it's the material under the lens it's getting pressed, and I worry if we go. Yeah, I would need to actually bring the slide even closer to that lens, and the lens. So actually, what happens is you have this can't see it very well, but I have four different lenses, 4x, 10x, 40x, and 100x, and the 40x has to be super close. And inside the lens, there's like these little cylindrical cases, and there's a lens inside them, and this lens is actually spring loaded in the bottom, and so as it comes up on the slide, it kind of presses into that cylinder, and it allows the lens to get even closer. Uh, and I need that in order to get the focal point that I want. But it's the slide cover is not going to be able to withstand the pressure that the microscope lens is going to place on it as I zoom in closer to it. And so I'm not going to try it. It just means this is kind of the best magnification we're going to get for tonight. All right, let's go back to 4x. We crushed whatever was under there, or definitely caused it to move away at the very least. I'd like to find just like one more thing that's not a worm. I want to find not a worm. Let's go into the more dense stuff. Like if you're a hungry worm, I feel like I'd want to be swimming around in here and just eating this buffet of junk. Hmm. Oh, hello. Neighborhood cat is on my front porch. Hello. Meow 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 meow. Meow 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 meow. Lucky. Meow 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 meow. Meow 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 meow. Nice. My cat Lucky saw the other cat. Oh, other way. Get over here. Oh, another worm. Going in. We're going to go down and to the left. We're going to chase it. We're worm chasers. Daylight comes in. Let's see. Oh, is it going over here? Am I predicting that right? Ooh, let's get back up there. All right, well, we're going to do one more sample. Then we're going to do one more sample, and we're going to call it quits. This is going to be the sample. It's going to have everything in it. I have a good feeling about it because I'm going to scoop up everything from everything. We're going to mix all of our samples together along with a very special mutagen that causes rampant uncontrolled chain reaction growth and mutation at an even higher rate than normal. And so within a few seconds, we should get something cool. I'm creating cancer. All right. Now, so we're going to take the sample from my yard. Oh, this sucks. I'm so sad that my dropper broke. Yeah, we're going to go all in on my yard sample. I feel like my yard sample is where it's at. Okay, how to get these out? All right, that's a little much. Let me get some of this stuff out of here. Accidentally dropped a giant item in here that we don't want because it's going to prevent the slide from closing and forming a good seal. Problem is, I don't know if I can get it off. Basically, a hole opened up in the side of my dropper, and now it's just like, yeah, I don't want any water. I don't want to suck up any water for you, which is severely dampening my plan. So I'll also get a better sample, better sampler, a better dropper next time. All right, almost prepared. Kind of stirring up the water a little bit, just getting some different color samples. All right, if there's going to be anything, it's going to be on this sample. So here we go. Let me find that slide cover again. Oh yeah, this is a very dense, very dense sample here. This is it. It's gonna be our last batch. So hopefully it's interesting. Wait, call it quits like in stream, or are you going to play a game? I'm probably going to call in stream because I have to work tomorrow and I have a presentation to do and just some stuff I need to take care of. And then I'm going to be gone from Tuesday to Thursday. So Tuesday, Wednesday, Thursday out. So I need to just get some stuff done for the night. And it's already 11:30 here. And I've been going on like 4 hours a night. I basically stayed up all night for the past 2 nights streaming and then hanging out with streamers and raid after raid after raid. And so I need to get some sleep. Uh, yeah, I gotta wake up tomorrow really early get the kids ready for school, uh, and then I just gotta get a bunch of work stuff done beforehand because my goal tomorrow is I want to take I want to take off a little bit early tomorrow since I'm going to be working all day from Tuesday, Wednesday, and Thursday, and I'll be away from home. I mean, I could maybe be convinced to do a Super Metroid arcade run, but no, I need to get to sleep. Also, there's some TV shows I want to catch up on. I want to watch like have you ever seen I Think You Should Leave, Mr. Fox? By Tim Robinson. I love watching a lot of like variety TV shows, and Tim Robinson has a new TV show out, and I've been wanting to watch. So I want to catch like 30 minutes of that before I crash. No, all right. I'll let me know if you like this kind of thing. I have a lot of these kinds of shows. Although my let's see, I think you let me put this in quotes so people don't think I'm telling you. I think you should leave. Uh, this is let's see by Tim Robinson. Basically anything with Tim Robinson. And yeah, what's going on? All right, we're 4X. Okay, we're just looking for any sign of motion, and then we're gonna zoom in on that motion. 8 hours of fixing stuff. Welcome to software development and configuration hell, where you regularly spend a whole bunch of unnecessary time doing the things you don't want to do. It's like, no, I just want to focus on my bot commands, not configuring it right. I feel like a lot of software and uh, well, a lot of software can be like that at times. There's an AI tool called Cursor, Cursor, and it's actually used as a programming AI tool, but it can do things like fix stuff on your computer. They could process the streamer bot code and the errors and tell you how to get it running. And it's an AI tool that can run locally, basically. And there's things like Claude Code, and we'll definitely talk sometime. There's some stuff that I use regularly to help me get unstuck with things when it's sometimes difficult to get somebody on the other line to like walk me through it. Yeah, no worries. We'll catch up soon. I'll be free next weekend too, so we can definitely chat. I think I'm free August 20 or October 19. Yeah, Halloween's next weekend. I have not. I have not yet. I came home and man, it's just been 24/7. I'll definitely check it out. Ooh, finally found something. There we go. Oh, a nice, maybe our best picture yet. Let's take a little snapshot of that. And I'm going to that's our picture. And then I'm going to save, allow. I'm going to call it microscopic worm 01. And let's throw it in pictures for now. We'll go back to our video and watch as a really primitive life form just flaps around as it's looking for food. And you can see its digestive tract is kind of swiggling back and forth. I re-downloaded the jar you gave me and tried to run it with the command. it through what was output into ChatGPT, and it helped me figure out some stuff. Okay, cool. Yeah, and I'll check it out too, because it's very possible that even though I'm running on this, Java Kotlin, which should run on any platform, it's very possible that your version of Linux or the jar I'm using is containing a library that doesn't run on your machine. Not for me, for you. Oh yeah, for sure. Yeah, I understand. And so what I'm saying is there's probably uh, it's probably something like that. I'm including something that I should have been a little bit more careful about including because it's not cross-platform. I'm checking out the DMs now. Oh, nice. Oh man, hey, thanks for sharing the information. And if you, yeah, this is useful. Useful. JetBrains Compose Desktop. And it was built with JetBrains Compose Desktop, but the native Skiko, okay, the native Skiko libraries weren't included in the build. Yeah, okay. Hey, this is super helpful, Mr. Fox. Thank you. I actually have that very same command in there. Uh, it was basically building a Debian file, but then I had an error when I was building it. I'll go through that. But yeah, this is the right approach, I think. Uh, mhm, mhm. Isn't ChatGPT great? Like, I know that's a lot of information, and it could very well be like right or wrong, or like partially right, partially wrong. But as an engineer, like having that level of information and things, ability to bounce ideas back and forth, like it's so great. Like that information you gave me, like that's why I use AI for everything. I get it. Like I know there's a lot of like controversial opinions on AI and things like that, but like I'm saying like it is super, super important or like helpful to just solve really any generic, like many generic problems, not just coding. Man, this thing is, we have such a good view of this worm. I'm very, very tickled that our last worm here actually looks good. Yeah. Well, that Debian file ultimately it's still going to package up and run the same thing. The Debian, that's just the package manager basically. So it's going to, like, if I put that file in a Debian format, then your Debian package manager, what you use when you do apt-get and install, like it's going to, or the dpackage command, like you're going to be able to install it. It's kind of like the app installer I use on Mac, but the underlying thing is still a jar. At the end of the day, no matter how we package it up and bundle it up, it's a jar. A jar is a jar. That jar is getting ran by Java, and it's either I pack up Java alongside my app, and which is I believe what I do, or you run the jar raw and using your system Java. And so, uh, but what you showed me was good because that tells me that I just didn't pull in the Linux library that you need. So similar to my Ninja Turtle game, which is also written in Kotlin. And oh, is our little worm breaking free? We can move to the left. Uh, but yeah, I have it produce both a Windows, Mac, and Linux executable. And I have to pull in different libraries for each of the operating systems in order to use the game engine. So, like, while Java itself is cross-platform, when you start doing things like game engines, you just invariably, oh, this is what I said earlier about like you'll find a lot of life forms on the side, and I kind of like it because they get stuck over here, so they're like a little bit easier to observe for a bit. One less direction that they can move in. Mm. So it's not that it's a yeah Mac OS jar file. A jar file is a jar file. It's just that it's a jar file, and it's missing the native libraries for your operating system. And so, but yeah, it's as you're saying though, it's a jar file with some other stuff packaged up for Mac. And normally you don't have to do that for Java or Kotlin. It just works without any extra configuration. But clearly I pulled in a library of some form without realizing it. It might be because it's an Android, I'm using the Android UI layer, and that might introduce some complexity. I really don't know. So what I'm gonna do is I'm gonna look through my code, and instead of like my solution to this is I just want to make sure I should even be whatever that library is. I want to know, should I be using it? What is pulling that library in, and is there a better alternative for cross-platform development? So I'm gonna do that research. It just like I don't want to just patch it or whatever. I want to like see like, is this actually a more serious issue, and I should be using a different library. And when I say library, I just mean like a collection of code. It's like one of those bundles of code I pulled in that Skiko library or whatever that was. I don't recall. Like, I'm actually not familiar what that is, so I can't speak to it. Usually I can, well, not usually, oftentimes I can see the error and just know what is missing exactly, but I'm not familiar with that specific library. Yeah, I think. Oh, and did you get it running? Were you able to find the libraries and get it running? Is that what you're saying? Or you just got or at least you understood what the error was with AI? Didn't? Yeah, yeah. I wouldn't try. That's what I was about to say. I was like, I wouldn't try because I think I'll be able to package that up for us with a little bit more ease. Just because, like, I think I just have to update my build script a little bit. And the first error I was getting though is it was like, we can't we can't run the package Debian command unless you're on Linux, which I'm like, okay. I build Linux executables on my Mac all the time. Okay, not all the time, but enough. So yeah, I just didn't. The error seemed strange to me when I first started looking into it, and yeah, exactly. You might get something working, but it'd be like brittle, and then the next time I give you an update, it could be problematic. Oh, all right. I guess today is just the day of the worms, man. Today is the day of the worms, where there's nothing else in our sample except worms and really, really small organisms that we can't really get into focus because they're small and fast. So I think we're just gonna enjoy our worm view for a few more minutes and see what happens. And I think we're gonna go check out Gameaholic and see what he's doing. Yeah, I'll tell you what. I'm gonna be sitting on my couch just watching some TV. So I'll look into this right now. I gotta. I'll figure out the timing, but I'm gonna look into this tonight. I'd love to get this resolved. Wow, look at that thing! Just it's incredible. All right. Well, I think I'm gonna call it here. We're hitting about 11:40 my time, so it gives me about an hour just to kind of chillax a little bit before I get to go to bed. So I'm gonna leave our worm hanging out here, and when I get off camera, we'll take him and release them back into their water sample so they can swim around and be happy worms as they swim through mud and devouring everything in their path. But yeah, let's go say hi to our good friend Gameaholic. Gameaholic 2D. I'm always scared to like to spell their name right. I wanted to get OBS set up as well, but spent 8 hours finishing setting up Letras and making sure things are working. Can't be broke by a random update I didn't authorize. Heck yeah, no, it's good to get that stuff all stabilized first. I agree. And yeah, I spent like hours getting this little layout. I also for the layout for the webcam. So there's actually a whole app here. I'm not going to like zoom out or whatever. There's a whole app with like all kinds of options for like zooming into the just changing like exposure times and white black balance, color adjustments, and all kinds of stuff. But like I had it, I just want it cropped like it is. And then I found this little wiki page here, or not wiki page, this Google page with some common samples for us, and or I could like turn on my webcam. Oh, double webcam! I have two webcams now, so to help with Rubiksing, and I don't really know why. And let's see. All right, with that, I am going. Oh, I didn't start the raid. Yeah, you know how to do the OBS stuff, right? Yeah, you've done that before. Yeah, I think it should just work on Linux. I would be surprised if like it doesn't. I will tell you though, if you have a problem, like if there's an option for like Linux video source or Linux, I don't know, call it Linux video capture. Sometimes those hang on me on Mac, and so I have to go into like the deprecated sources, and I use like the deprecated window and stuff. But yeah, I don't know. Let me know if you have any issues on the Linux side there. Although you seem pretty capable of figuring out your own problems. So I don't know if that meant to, if that sounded uh like I was implying you can't. So I didn't mean that. Uh, I still need to check out Pop OS too. Yeah, I'd seen Pop OS more and more recently, and I haven't used it. Yeah, so Kubuntu was what I used Ubuntu, and they used to have a desktop operating system, and then they switched to that more mobile OS where it was like a tablet, and I was so bummed out about that because I was like, no, I want a freaking desktop. And so then I looked into Kubuntu. Ultimately, I went with Linux Mint, and it gave me what I needed. And but then they broke internationalization with the SCIM to iBus transition for multiple years, and I was just like, I had to leave Linux because I use international language, like international text, a lot with my wife and friends, and I needed that as a function. And then, but it seems like they've gotten a lot of that taken care of, so it's pretty sweet. All right, guys, let's kick off this raid and go say hi to Gameaholic. And dun, 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 dun. All right, peace, everyone. I promise the next the next Microscope adventure is going to be filled with a lot more life. I'm going to make sure of it. All right, peace and have a good night, everyone. And yeah, yeah, send me that invite, Mr. Fox. And thanks again. Thanks for hanging, man. It's good to see you as always. And yeah, we'll reach out and DM, and we'll get that Super Metroid tracker running on Linux. I promise. All right, peace. And